Welcome to our Kentucky Science Center Virtual Youth Science Summit. This is our third annual one. We're very excited to have you all. So I would like to say that we've been doing our Youth Science Summits for over 10 years, and we're very excited to introduce you to new STEAM careers and kind of learn about what people are doing around Kentucky. So, but before we get things started, I want to kick it off to our CEO, Mike Norman.
Thank you, Veronica, and good morning. Welcome to your Kentucky Science Center Spring 2022 Youth Science Summit. I am so glad you joined us here today. You know, we have almost 500 people with us today from our friends all over the Commonwealth and the region, from over in Moorhead, over in Eastern Kentucky, down to Logan County in the south, all the way up to Columbus, Indiana in the north. I am so thrilled that you're with us today. And I want to thank our partners who make this possible. Their support really helps us do these virtual Youth Science Summits. So thank you to Kentucky EPSCoR. Thank you to Toyota Motor Manufacturing of Kentucky. And if you know anybody at one of these great organizations, please thank them personally for us. So we have a fantastic lineup of speakers for you today. They're going to share with you the amazing work they're doing in STEAM fields. So my challenge for you is this. While you listen, dream about what lies ahead for you. You know, the possibilities are endless. The STEAM careers of tomorrow, they don't even exist today. So it's up to you to make the future what you want it to be. And I'm so confident that you will. So again, thank you for joining us. Have a great day. Enjoy the time. Thank you.
All right, thank you very much, Mike. So again, we are going to get started with our keynote speaker. Dr. Holm is from the University of Louisville, and she is the associate professor at the Christina Lee Brown Envirome Institute. She specializes in water and sanitation, environmental health to improve the well-being of the most vulnerable populations. So we're going to kick it off to her. And please, if you have any questions, we're going to talk about it after her video and then get those questions to her so we get your responses. Go ahead and kick it off.
Hello, my name is Rochelle Holm, and I'm an associate professor in the Christina Lee Brown Envirome Institute at the University of Louisville. Thank you and good morning, everybody. Welcome to our third virtual Youth Science Summit. What a great day we have in store for you. I am so glad that you are joining us. Today, we are going to see participation and colleagues from around the state. You're going to be inspired by listening to some great STEAM professionals share their stories and show you what the possibilities are and what the future can hold for you. It doesn't matter where you are in your career thinking. Science, technology, engineering, math, and art are fields that you can enter. We are able to offer this program at no cost to students because of grant sponsorship and funds from the Kentucky NSF EPSCoR and Toyota Kentucky. I would like to share a little bit about my own science at the University of Louisville that has been used the past 2 years during the pandemic. Countless times I am asked, wait, you are a scientist and you play with what? That is because I'm part of a team that tracks what you flush down the toilet as part of sewer tracking to monitor COVID-19 across Kentucky. Sewer tracking requires a team of environmental scientists, laboratory technicians, and the local sewer utility workers in a strong STEAM collaboration of professionals together as one team. I have a background in environmental health, and specifically water, sanitation, and hygiene to improve the well-being of the most vulnerable population groups. And part of this is the role of fecal waste streams in public health surveillance. The team at the Envirome Institute is leading the co-immunity project for severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, which is the virus that causes the COVID-19 disease. Tracking community feces for health is not new and has been used across the world for a wide range of public health topics, including identifying the use and changing habits of illicit drugs, global eradication of polio, and pathogens. The University of Louisville is just one of 3,000 sites in 58 countries across the globe doing this work of tracking COVID-19 from the sewers. Instead of taking individual stool samples, such as at a doctor's office, by taking samples out of strategic zones in our existing underground citywide sewer lines, we are able to collect a stool sample that represents many individuals and households as an anonymous glimpse of what has gone down the toilet in different zones of our community the last few days. Many, but not all, COVID-19 infected patients shed SARS-CoV-2 in their stool, and this shedding can last more than 3 weeks, so it gives us a nice wide window to capture a positive sample. And because SARS-CoV-2 can be shed in the stool of individuals with symptomatic or asymptomatic infection, wastewater surveillance can capture data on both types of infection. So you might ask me next, well, how does this actually work? In Jefferson County, the sewer system contains active elements that have been in operation for over a century and combines rainwater runoff, industrial wastewater, and household sewage into the same pipes among most of the network. Our research utilizes the existing sewer system for pandemic monitoring. Well, the University of Louisville has selected zones of 8,000 to 350,000 people where everything that goes down your toilet, sink, or shower is combined, and we collect a small amount of that sewer content weekly. We have a range of sample collection points, from neighborhood level manhole access points to large wastewater treatment plants. We work in collaboration with an amazing team from the Louisville Jefferson County Metropolitan Sewer District to help us collect the samples, and then all the analysis is done at the University of Louisville labs. We started collecting sewer samples in May of 2020. We currently are looking at 11 sewer zones, which cover most of Jefferson County, and each of the 5 treatment facilities operated by Metro Sewer District for a total of 16 sites. Sampling personnel from MSD set up a portable, automatic 24-hour sampler for us, a little bit like a robot. You may have seen one out in our community. Tubing reaches down into the sewers to collect a small volume of sewer water every 15 minutes over a 24-hour period, and then it's analyzed at the University of Louisville. We have collected over 2,000 wastewater samples in Jefferson County, and it has been a lot of work. Our findings in the sewers are that every site shows the presence of SARS-CoV-2, regardless of the area of the county. We see the virus across income and ethnicity variations. Together with county-level clinical data, our sewers are helping us get a better picture to understand infection rate changes in large population centers of Louisville. As more people get vaccinated, we expect our work to soon transition to following decreases of zones within Jefferson County. We meet weekly with our local government public health officials because wastewater data should never be used on its own. Rather, it provides another data point for consideration alongside other clinical data, such as positive case rates and hospitalization rates. There is still a lot of ongoing research in this field. While the trends are often consistent, the direct relationship between wastewater SARS-CoV-2 concentration and COVID-19 infection patients is still less clear. One of the biggest benefits of the added power of community health via sewer samples was during the recent Omicron wave, where more and more people started to move toward at-home testing. Many were not officially reported to state or national surveillance. Wastewater monitoring equitably captures cases as people flush their toilets. And because we run genomic sequencing on all of our samples, we can see what variants of COVID-19 spread in close to real time throughout the county. Throughout the program today, I hope that you are inspired, that you will learn something new, and be willing to break STEAM boundaries in your community, in our state, nationally, and internationally. And know that STEAM is a part of all of us also as we flush the toilet. I do want to say thank you to all of our STEAM professionals for partnering with us being here today. I want to thank also our amazing team who put this together. There's a lot of work that goes into today. And finally, I want to thank KY NSP EPSCoR and Toyota Kentucky. Without them, we wouldn't be able to do this today. Welcome to our program.
All right. So please, again, we want to encourage questions for our program today. So we're going to be using the Q&A feature, and we will be able to send all of your questions to Dr. Holm in order for you guys to learn more about this great project. So do we have any questions for the group? I think it's fascinating to find out how all the different things are connected, and I can't wait to learn more. So if you think of questions, please utilize the Q&A feature. And so that way we can get our questions to our next speakers. We'll give about 15 seconds for Q&A for the speaker. And if you did sign on later, we will have this available on our YouTube page. It is currently streaming live, so you can always rewatch if you missed any of that video before. That way you and your students can, teachers and students can come up with questions that we can ask. And if there are no questions, I can go ahead and introduce our next speaker. So Matt Kolmann, he is going to be actually going to be talking about piranhas and their cousins. I'm very excited. Actually, my background's in marine science, so I might have to ask some other questions too if students don't have any. So, but go ahead, Matt, take it away.
Great. I just need to be able to share my screen and let me know, and then I can get started.
Okay, you should be able to do that now.
Perfect. Thank you. So hello, thanks for being here today. The title of my talk is Fishes, Forests, and Fruits, How Piranhas help the rainforest. And again, my name is Matt Kolmann. I am an evolutionary biologist at U of L. And what my work and my lab does is trying to understand the roles or jobs that animals fill in their environments and how they evolved to get there. And I work predominantly with fish, what zoologists would call the model system that I work on. Now you might ask, why fishes? Well, there are 35,000 species, and so for comparison, there's around 6,000 species of things like dogs, cats, or cows. So in fact, if you tallied up all the vertebrates that aren't fishes, so the reptiles, birds, and things like that, you'd still have more species of fish than anything else. And this diversity gives us a lot to choose from with respect to what we want to study. And so I always tell my students, if you're interested in something weird that an animal does, any kind of behavior or trait, there's a fish that does it. And so we've got fishes that climb, we've got fishes that glide or fly. We even have fishes that walk along sort of the bottom of the sea. And there's lots of fishes that do these weird things too. So not just one or two species, but potentially tens or more. And so, for example, let's say you're interested in something truly weird, like why you would have a saw blade of teeth on your face, which is what this sawfish has. Where does this kind of trait come from? Why does it evolve? Well, this weird structure has evolved 3 times, once in sharks, twice in rays, but one of those groups of rays has actually gone extinct in the far past. And so this structure, which is evolved from highly sort of changed scales along the nose, and those scales look like teeth, is used to capture and stun prey, and also to defend this fish from things that might try to eat it. And so the research that we do in my lab is interested in these sorts of crazy traits because it lends insight into how the animals engage with their surroundings, what they eat, where they live, how they use tools like teeth, jaws, and fins to get to where they need to be and eat the things that they need to sustain them. And so I use this manner of inquiry to understand the roles that the animals play and how this explains why there are so many different species. And so where does the biodiversity that we have on Earth come from? But most people don't really appreciate fishes like I do. And so I like to start with kind of an example that's probably closer to home. So everybody at some point in their lives has seen a show on PBS or Discovery, something like narrated by Attenborough, and 8 times out of 10, it's about the mammals we find on the African savanna. And so you've got lions, the predators, the antelopes, the herbivores, and hyenas, the scavengers. And although we know this is a little simplistic, right, because hyenas often hunt and kill their own prey, and lions in turn sort of steal the prey from the hyenas, I digress by I'm firmly in team hyena. But in this case, we have three different families here: cats, hyenids, and ungulates, filling three distinct roles. But I study fishes. So who's a good group to study to understand where ecological diversity stems from? And so I would say that piranhas are a great group. So I love piranhas and their herbivorous cousins, the pacus. And what's so cool to me about these fishes is how many jobs they fill in the Amazon. And so if you go to the Amazon in Peru or Colombia, like for example, you'd find piranhas acting as top predators. You'd see them behaving like scavengers, so sort of cleaning up dead and dying things. And finally, you'd find pacus eating plants, seeds, and things like that. Heck, there are even parasitic piranhas that just eat the slime, scales, or fins of other kinds of fishes, which to this day just strikes me as really, really weird. And so this is a CT scan that's on your right. And within the stomach of that fish, there are 16 of these scales. And each of those scales is 1.5 times the size of the eyeball of that fish. So this species actually specializes on feeding on fish with particularly large scales, which is just really, really weird. And so I'll point out that in this one family of fishes, compared to those three families on the savanna, we have all this diversity, which begs the question, why are piranha and pacu-like diets so diverse? And so this is one of the things my lab does, is try to determine why there are so many species of fishes on Earth and why those fishes are so diverse in terms of the jobs that they fill. And so this is a family tree for piranhas, and it's color coded according to what they eat, which we'll discuss in just a bit. But first, we need to be able to sort of break down what this family tree shows. And to read this is fairly straightforward. So think about this as kind of the hereditary tree you might hear your grandparents discussing, but instead of aunts and the uncles, we've got species on the right, A, B, and C. And they're connected by these lines which show which species are more closely related to each other. And so in this case, B and C are closer related to each other than they are to A, but all three form a group. And the ancestors of these species are denoted by squares, right where the lines come together. And so the ancestor of B and C is this first black square in the middle part of your screen. And we would call this the most recent common ancestor for these species, or the MRCA. This also means that more recent time is towards the right and older events are on the left. And usually the length of these lines, or the branches of the tree, tell you something about how old these groups are. So if we apply this to a really simple tree for pacus and piranhas, we can see that piranhas split from pacus around 15 to 20 million years in the past. Now I've expanded this to include all species of pacus and piranhas and color coded it according to diet, so according to what they eat. And we see that these fishes do all these different jobs, from eating fruits and seeds to consuming plankton or eating smaller fishes. And if you look really closely, what's really kind of strange here is that eating fruits and seeds has evolved several times across this one family, at least 5 times, we think. And it's even evolved in piranhas, which might come as a bit of a shock, that there are the vegetarian piranhas, right? That vegans have infiltrated even piranhas. And in fact, about a quarter or a third of piranha species will eat fruits and seeds. And heck, the ancestor of all piranhas that still exist today ate fruit. Now, where did this come from? Why is this the case? Well, fruits and seeds are plentiful in the Amazon, where trees might produce fruit year-round. Their fruit are especially plentiful during the wet season. And unlike fish, you don't really need to chase fruit, right? So you'll find pacus and piranhas with their guts packed with seeds and digested fruit. And this makes sense. They've got a resource that's all around them, and it's not hard to find. In fact, some plants have evolved fruits to attract the fishes to eat them, like how flowers evolved to attract pollinating insects like bees. And as it turns out, these fishes, were probably the earliest fruit-eating things in the Amazon. So before things like monkeys and birds, these fishes evolved around 40 to 50 million years in the past. But why is this important at all? And we need to sort of take a step back away from the fish and think about the plant that produces the fruit. So plants produce these fruits that attract the fishes. The fruits are sweet, they float, they fall into the into the stream, and you'll actually find groups of these fishes that stake out one tree for weeks, waiting for the fruit to fall. And so plants make a gamble that the fishes will eat the fruit and sometimes travel hundreds of kilometers away from the parent plant. And so the fish is dispersing the seeds away from the parent plant to new places where that seed can grow. So plants make a wager that the seeds won't be destroyed and that the fishes will poop out seeds as they digest the fruit. And in fact, the tie between fish and fruit is so close that some seeds won't even grow unless they've passed through the gut of a specific species of fish. And so by dispersing seeds away from the parent plant, this limits the chance that the parent plant and the seed will have to compete. And this directly relates to the health of the rainforest and its growth. But why should you care? So many of these plants have use to us and the folks who live around the Amazon. For example, if you use a loofah to scrub yourself in the bath, those plants are spread by fishes. And so a slew of plants which have medicinal and industrial uses are actually distributed by plants. But you might think that I was also concerned about the tools that the animals use to do these jobs. So how do piranhas and pacus eat fruits and seeds? Well, they're not like other fishes with these weird jaws that kind of shoot away from their faces. Piranhas have jaws and teeth that are like yours. They're firmly attached to the base of the skull. So how do the pacus and piranhas eat fruit? Well, they bite it. They bob for fruit from beneath the water. And so pacu and piranha teeth are actually adapted for shearing through prey and even crushing the harder parts of food like seeds. And so they have two sort of things that aid with this. The first is that pacus and piranhas replace all their teeth, upper and lower, at once on one side of their face. So around 3 times a year, they'll lose all the teeth on one side of their mouth and then replace those teeth from teeth that are developing within the jaw. So again, kind of like you. And this helps piranhas and pacus to keep at least one continuously sharp set of teeth around. So piranha teeth also actually lock, and this helps them to sort of act like a singular saw blade to help them shear through prey. And so we've been actually seeing how good these teeth are at sort of puncturing and destroying fruit. And so we've done this by CT scanning piranhas, 3D printing their teeth, and then driving those teeth into different fruits to understand how tooth shape predicts how well they behave in destroying that fruit, destroying the cells, the pulp, and the rind. And so it turns out there's a lot of similarities in how fruit-eating fishes and things like monkey teeth are shaped. But why would we be concerned about this too? Well, there was a fossil piranha that was found called megapiranha that lived 20 million years in the past. And this fish was the size of you. And up until now, it's been assumed to eat meat like other, but not all piranhas. And so the testing that we've done has found that the teeth of this fossil species of piranha behave more like the piranhas that eat fruit than the ones that eat meat, suggesting too that this species might not have eaten meat or might have eaten meat and plants too. And so where does this kind of end with us? Well, pacus are incredibly important food fishes in the Amazon. And so we need to harvest them sustainably in order to balance the service that they provide us, which is delicious food, and the service that they provide the rainforest, which is dispersing seeds and fruit. And so what did we learn at the end of this talk? Well, there's more species of fishes than there are any of the other vertebrates. Pacus and piranhas eat a diverse sort of set of prey. That these fishes are some of the primary seed dispersers in the flooded forests, and that the rainforest health is probably more than just trying to protect the trees, but also protect the things that serve the trees, like the fishes. And finally, we found out that robust teeth help these species to kind of eat fruits, tear through flesh, and crush seeds. And with that, I'll thank all the folks who have funded me in the event coordinators today. And with that, I'll take any questions.
Excellent. We do have some questions. That's fascinating. So the first question, do most fish only eat a couple different things? You talk about how different, piranhas are, where they can be eating just meat or kind of a combination, but is that common in most fish?
Yeah, so it really depends, right? There's a lot of different species of fishes, and a lot of them do really, really different things, right? So there's that one scale-feeding piranha that literally really only eats scales and slime. But then a lot of species, like I used to study stingrays, eat kind of like a hodgepodge of things. It's like you go to a buffet and they'll take advantage of anything that's there, from shrimp to fish to insects to clams. So it really just depends on the species. What's so cool about this group is that they specialize, a lot of species specialize on one kind of prey to the exclusion of the rest.
Excellent. I have another question from the group. What does sparkling fish sustainably look like?
That's a really complicated question, but it's a really important one to think about. So one thing you always want to do is kind of get an idea of how much fish is used in a particular place at a certain time, and which species are being used, and make sure that you're never kind of harvesting them over their ability to be able to grow back or bounce back from how much you take each year. So you need to really consider the biology of the animal, how many young it produces per year, how often, or what proportion of those young survive from year to year. But you also need to balance the importance that those food fish play in, the diets of us too. So it's a really, really complicated thing.
We have another question from a student, but I wanted to ask, what got you into piranhas?
I was doing field work in the Amazon and realized that people didn't really consider how diverse the diets were of those things. It kind of blew my mind when I first learned that a lot of piranhas eat plants, and it was sort of strange that nobody had considered how that had evolved or why it had evolved. So that's what got me started.
It is fascinating. All right, another question we have: do piranha teeth resemble human milk teeth or shark teeth, which have been compared to scales? I think I have that one right.
Yeah, that's a really interesting question. So piranha teeth are kind of more like true teeth, and I should back that up a little bit by saying there are many tooth-like things called odontodes, and shark teeth and shark scales are technically built of the same stuff, right? So they're built out of like enamel and dentine. And so if you were to ever touch a shark, you know how it feels like really, really rough and sandpaper-like? That's because they have basically teeth that form their scales that are on the outside of the shark. So piranha teeth resemble that in that they're made from the same stuff, and your teeth too are made from the same things. And so the interesting difference there is that shark teeth are kind of replaced kind of one at a time throughout their life, but they could go through thousands of teeth throughout their whole life, and sharks live a long time. Piranhas do something a little bit more distinctly where they actually replace all the teeth on one side of their face at once, and those teeth develop within the jaw, whereas the shark teeth kind of develop within the skin on the side of the jaw. So piranhas in some ways are a little bit more like your teeth than they are like shark teeth. But it's interesting you saw that because people originally thought that piranha teeth behaved more like shark teeth. And that's kind of explains why they always want to have a sharp set of teeth like all the time. So great.
Fascinating. I'll give more time for a few more questions, please use the Q&A for that. I do have another question of my own. So if someone was really excited by learning about piranhas, what are some of the next things that they could do in order to get more experience, get more exposure? What are some options for students?
Yeah, so one thing is, to always get involved with the zoos and aquariums that are nearby. So like the Louisville Zoo is a great place. I'm not sure if they have any piranhas. I think they actually do. So it might be good to reach out to them and see what you can do to help out. Piranhas are banned in the state of Kentucky because we're concerned that they might get loose. So you can always come and write to me, and we might have space, for even the high school students to come and work with us. We won't have piranhas yet, but hopefully soon we will.
Okay, so again, this is something I'm really excited about, so I keep coming up with questions. What about like invasive species, and does your work have anything to do with thinking about invasive species?
Not yet, but we're actually going to be doing some of that in the near future. So I also work with snakeheads, and snakeheads are an invasive species within the US. We don't really work on the invasive ones, but we work on how that group evolved and what makes them so good at like invading from place to place. There aren't a lot of invasive there aren't a lot of invasive piranhas within the US. There may be some in South Florida, but pacus are very invasive in South Florida. We don't know to what extent, and again, I'm not really an expert on that part, but because Florida is a tropical place and things seem to be warming up, there's great potential for harm if they get released.
That's great. Any other questions for Matt? So, we have another one. So this question is, we have a lot of youth here today. What made you decide to do ecology for a living? And what does that look like as a career path?
Yeah, so great question. My parents kind of doomed me on this one. I had a fish tank in my room from before I was born. So I got into this from the pet trade, and I was interested in the management of fish species, and then sort of changed directions like midway through. So I used to work on sharks and stingrays, and still do to some extent. What does that look like as a career path? You're going to be in school for a really long time. So if that appeals to you, you should think more. You should think more about it. I've been able to travel to some really incredible places and work with some incredible people, but it really is a long career path. But it's an absolute blast. We have a lot of fun. I do want to say, if I have a little bit of time, is that okay? You might have heard that I have a stutter, that I stammer, and I just want to let you know, if anyone wants to talk about or ask me about what it's like to not be able to hear or speak well within the sciences, to please get in touch with me.
That's awesome. And so yes, all those who are participating, we will share out all contact information from each of our presenters. So that way you can either reach out to us to get to them, or you can reach out to them directly. So it's an excellent point. So we have another question. Are piranhas banned in Kentucky because they are invasive or because they are threatening?
That's a really good question that I don't know the answer to. I am 99% sure that piranhas do not exist in Kentucky, meaning they haven't invaded, right? So I think probably the threat of invasion is what is most concerning there. And so you should be able to check that if you check with the state and check with Fish and Game. But yeah, I'm pretty sure there's no invasive piranhas within the state.
Knock on wood. All right, any other questions? And Matt, if you want to hang out for just a little bit, just in case one pops up, that would be
sure.
Oh, we got another one. All right, so do piranhas only live in saltwater, I guess? Or
so actually they are only in freshwater.
Fresh, all right. Excellent. All right, final thoughts here before I introduce the next speaker. Well, thank you, Matt. That was a lot of fun. Again, I'm a big fish nerd, so it's great.
Cool.
And because my undergraduate was in marine science, and everyone thought I just studied mammals. That's all I'm like, no,
yeah.
not the case. So
Yep.
if someone is really interested in marine science, go for it. It's awesome, but you're going to be learning a lot more than just about sharks and dolphins. So, all right.
Thank you.
All right. So our next speaker is Jay Stotman. Are you there, Jay?
I'm here.
All right. Let me see. Let me make sure you can share screen. Okay. So let me do a quick introduction. He's going to be talking about the science of archaeology and how we can use archaeology to kind of learn about our past and help change our future. So thank you very much, Jay, for being here.
Great to be here. Get started here just a second. Okay, sorry about that. All right, my name is Jay Stotman. I'm an archaeologist with the Kentucky Archaeological Survey, which is based out of Western Kentucky University. We do archaeology in Kentucky. Believe it or not, there is lots of archaeology in Kentucky. We mostly do work around the state, and we often work with the public and invite the public to work with us. I'm going to be talking to you a little bit about the science of archaeology and how archaeology uses the process of science, and give you an idea about one particular subject that we might work with, because there's 10,000 years worth of human history out there, and we can investigate all that with archaeology. But I'll give you one little aspect of history that is very connected to our present. So the idea is that we're not just always looking in the past, we're also looking towards the future. So what do you think of when you hear the word archaeology? Perhaps you think about something very exotic like the Great Pyramids in Egypt. Or if you're old like me, you might think of Indiana Jones. Or dare I say, you might think of dinosaurs. But archaeology is a process. It's one way that we can learn about human history and culture. And the operative word here is human history. So we're interested in people, and that does not include dinosaurs. It is the study of human-made objects and their context. And so I'll take a few minutes to explain what I mean by this, and how we can learn about history and culture from basically things that people threw away. And it has a lot to do with where we find them. So the process of archaeology starts, like most science, with a research question. We just don't go around the countryside looking for treasure and old things like that. We do archaeology because we want to learn something. There's something in particular we want to know about. So that is our research question. And every archaeologist starts with one of these. We have to have a plan for answering that question. So where are we going to go to find the information that we need to answer our question? How are we going to get that information? And how long is it going to take? How much does it cost? All these things we have to think about and plan for when we start to do archaeology. And then, of course, we have to collect our data. So there's a lot of different ways that you can collect data. I'll talk a little bit about that in a minute. But it's all about data. So we can't answer our questions if we don't have the data to do that. Now, of course, after we collect our data, we got to analyze it. So what does this data tell us about people in the past? How does it help us answer our question? Does it create more questions? So we have to analyze that data, and then we interpret it. So what did we learn from that data? Did we answer our question? Did we create 50 more questions, which is usually the case? What can we say about people in the past? And of course, more importantly, we need to disseminate our information. We need to get it out to the public. We need to get what we learn out to everyone else so we can share what we learned. All right, so let's talk a little bit about research questions because it has a lot to do with what we study in particular. So research questions are basically what we want to know about culture and history. The thing about culture and history is it's very complex, but we can use archaeology to help us better understand how history is created, what it was, and learn more about culture in the past so we can learn kind of where our culture and history comes from today. So our questions can be very simple, like, is there anything there, right? Is there anything at that place? What used to be here? Or it can be very complex, such as understanding people's economic status and their identity, race and ethnicity. These very complicated issues that we're used to thinking about today. These same things were going on a long time ago. And we can understand these complex concepts through the things that people left behind a long time ago, and connect those to how we got to where we are now. So the data that we work with is varied, and all the examples I'm going to show you today are actually from Kentucky. Our main source of data is what we call artifacts. Now, an artifact is basically anything that was made or used by people. It can be from 10,000 years ago, 2,000 years ago. It could be things like this 10,000-year-old spear point, or a battered rock, pieces of flint, a weird-looking rock, fired clay pots, or it could even be from 150 years ago, like these fancy pieces of dishes, marbles, and glass bottles, bone-handled toothbrush, and all these bits and pieces that we see in the middle of the screen there. However, those artifacts have very little information to give us if we don't know its context. So context is the single most important source of data that we use. So we need to know where the artifacts were found and what they were found with. It is the context that allows us to interpret these artifacts. Even though we may know what they are, we may not know how people use them. We may not know what they meant to people. But we can't do that without the context. So most of my work is really focused on measuring and locating where artifacts come from, what layer of soil it was in, how deep that was, where it is spatially on the landscape. So basically, archaeology is about people in the past, and people live here for 10,000 years. And I mean here, Louisville. All right, so we may ask the question, why do we need archaeology to learn about a time when we had photographs and written documents? Because if we're trying to learn about people's history, and we're learning about people who have recorded history, how does archaeology really help us with that? Well, this picture that you see right here will tell you everything you need to know about that question. So when we talk about people in the past, we're talking about all people in the past. So not everyone in this picture is equal, and not everyone in this picture's their histories have been told or we know very much about. So when we think about people, we think about this broad group of people who have different lives, different statuses, different conditions, and not everyone's story is always told. So there were people who were not written about, or people who could not tell their own story. So how do we know where, how they lived, what their culture was like, and how does that relate to us today? How do we find out where they lived? Oftentimes the places that the people we don't know much about lived and their lives are literally erased from the landscape. They're removed, and all that's left is what's buried beneath our feet. So we can take things like broken bits of glass, nails, stones, patterns of stone that we find in the ground, and we can reconstruct what a building might have looked like, how big it might have been, and get an idea of how it was used and how it might be furnished. So what you're seeing here is a digital reconstruction of a building, a slave house that we excavated at the Farmington historic home. And we're able to do that based on the data that we found. We also find things that are more personal, such as this commemorative George Washington coin that was turned into a necklace or a pendant, or what we consider a charm, depending on where it was found. And this one happened to have been found at a slave house site. We also find everyday items that can not only tell us about things like what people ate and what kind of possessions they had, but there are also things that with these everyday items that can tell us a little bit more about people that we might not think of. So when you look at these pictures, there are a couple of things that are these things have in common. One of the most obvious things you might see in the pictures I've selected here is that they all have an X scratched on them. The other thing that's similar about all these objects is their context. They all came from a place where enslaved people lived. Not the same place. In fact, these objects came from 3 different historic plantations in Louisville. So these things are similar in the respects of there's a pattern in the objects themselves and their context. So what could this mean? Now, one thing about doing archaeology and interpreting archaeology is exploring a lot of different options, a lot of different interpretations. So our first thought with something like this might be, okay, so these are objects that were owned by slaves or used by slaves, enslaved people. And because enslaved people didn't have any freedom, because they were enslaved within a very oppressive institution, these objects must have been given to them by the people that owned them or enslaved them. And so thus the people who gave them these objects X'd them out so it could be easily identified as something that they can have. But if we think about that, culture is resilient and culture is important to everyone. We can think about this in a different way, because if we think about the context, these were things that enslaved people used. We think about what these X's might mean, and also think about the people who use these objects had some type of agency, in the sense that that their whole lives weren't controlled, that they were actively doing things to make a life for themselves. If we explore this a little more, this can tell us something about enslaved people, something a little more empowering. So we think about that. these X's, archaeologists have studied these and said, okay, well, what about the people who enslaved people who might have used these? Where did they come from? And what could these things mean? And so they went and investigated religion in West Africa and found that that West African religions used what's called a cosmogram to define their world. So you can see an example of this on the screen. Is that it shows the life cycle, birth and life and death, rebirth, the importance of air and water, reflective surfaces, in this shape. And so if we think about this cosmogram and abbreviated forms of this cosmogram, you can see where something like a scratched X might be representative of a cultural tradition. We also see, if we take that a little bit further, we have also seen where at enslaved people's sites, we have found what could be interpreted as an ancestor shrine. Now, ancestor shrines are common forms of beliefs and practices that people of West Africa and in their religion often use. They use it to communicate with their ancestors and honor their ancestors. And we found that there are there's a lot of similarities in some of the things that seem very innocuous that we find in archaeology, but are very similar to ancestor shrines. So at one plantation in Jessamine County, we found this ceramic plate that had these objects underneath the plate, and then they were all buried together. Now, these objects don't seem like they're all that distinctive. They're just basic kind of objects. But when we think about them in the context of West African belief systems, we see elements of an ancestor shrine. The color white is very important. The connection to water with the freshwater snail shell, and these metal objects, brass and iron, are common elements of ancestor shrines. So what this tells us is it tells us that enslaved people were actively fighting against the conditions in which they were living in under the institution of slavery. They were fighting to keep their traditions and honor their traditions, and creating culture, as well as communicating with other enslaved people on other plantations. So this gives us a different perspective on enslaved people being powerless victims of slavery to people who are resilient and people who are fighting for scraps of humanity and trying to keep their humanity and their traditions alive while making new cultures and new traditions, all under an oppressive system. And this gives us a different perspective of people today. So what can we do with this information? Well, we can start to fully recognize people who were forgotten in history or whose histories were repressed. We can start to tell forgotten stories or repressed histories, start to tell those stories. We can even take part in the recovery of lost and repressed history. So people can come and work with us and touch and find those objects that were lost 150 years ago, and see things that people haven't seen in a really long time. And a more recent project, just a couple of months ago, we were doing ground penetrating radar just near the Oxmoor Mall, which used to be a plantation, and finding what we believe is a cemetery for enslaved people, a place that had been forgotten, no headstones left, kind of crammed, or has all these buildings developed around it. So we can locate where people were buried and bring that history back to life. So it's just like all other kinds of science. The more information, the more data that we get, the better our interpretations will be. So no scientist goes in thinking that they have the answer to everything, and they understand that as more data becomes available, our interpretations will get better. All right, any questions?
Yes, thank you. We do have some questions here. So the first question we have is, what do you think future archaeologists will study from our current culture? And what conclusions do you think they will draw?
Yeah, that's a great question. Actually, there are archaeologists who study our culture now. So artifacts we tend to think of as being very, very old, but an artifact is anything made or used by people. So the things that we throw away now tell us much about ourselves. So there are archaeologists who have studied landfills, and that information has helped people understand what we throw away and how we throw away items, and what do we do with those things. And so there are real questions that archaeologists can study about our own culture through archaeology. I myself, I've done a study of litter in my own neighborhood to learn how we might be able to combat that problem. And I guarantee you, if I came to your school and looked through your trash, I could probably tell a lot about you all too. But it's not always about the past. Archaeology is one of those things that can continue. It's a process of learning about things.
Okay. I have a follow-up one for myself.
Okay.
What got you into archaeology?
Archaeology, what got me into it was I was just always fascinated by history, and I always liked to be outside. And that's the great thing about archaeology is you can be outside. There's a lot of diversity in things you do. Sometimes I'm in an archive doing research. Sometimes I'm in front of my computer writing reports. Sometimes I'm out in the Danube National Forest. Sometimes I'm at a really cool old historic site. So for me, I just think it's a really diverse kind of fun activity to do. It's just really interesting. I've always been fascinated by people and people in the past.
All right, thank you. We do have another question here. Do archaeologists work a lot with people who study anthropology?
Actually, we are anthropologists. So my degrees are actually in anthropology. Archaeology is a subfield of anthropology. So we do the same things as anthropologists. We just use different data, and sometimes the time period in which we study is different. And I work with anthropologists all the time who study more recent cultures. So we're pretty much one and the same.
I like this question. How does the internet impact the study of our current culture? What are your thoughts on that?
Yeah, I think from an anthropological point of view, that would be very interesting about that. I think that goes a lot to the question of what is data, and what and can we trust a lot of the data that we see out there? But social media and the internet are actually tools that we use as archaeologists. It helps us better understand our material culture, and it connects us to cultures and ideas that might have been a little more difficult for us to access. But yeah, certainly one question I've asked about our social media and things like that is that these are our aspects of history. These are recorded bits of history, and what will we say about that? And how permanent is it? Does it last forever? It's not anything we're going to be able to dig up, and as far as I know.
I guess it'll be like, computers somehow, but yeah, that's not quite the same thing. Are there any other questions? We'll open up for a couple more minutes for final questions. I guess, again, I see that you can do a field trip. So what are other ways that people can become involved?
I work at a place called the Farms and Mormon House. It's Riverside, the Farms and Mormon Landing. And we've been doing a field trip program out there for about 25 years. And we also have events throughout the year where we have the opportunity for people to come and dig with us and participate in the programs. So right now we're doing washing of artifacts and cataloging. We do that twice a week. We will be doing the Cultural Pass program during the summer. So if any of you guys do the Cultural Pass program, you can come out and visit us at Riverside and do some digs with us. And then there are lots of other places. If you want to search the Kentucky Archaeological Survey, we can connect you to a lot of other things about archaeology and public opportunities.
Excellent. Oh, another question just popped up. How competitive is the field? It's a good question.
I guess it's like anything else. I mean, there are lots of career opportunities in archaeology. I mean, a lot of times people think that your only career choice is to be a college professor somewhere, and that's really not where most archaeologists work. There are many archaeologists that work in the private sector, work for governments, managing archaeological sites and cultural resources. There are private companies that do archaeology because a lot of development requires archaeology to be done. So there's a really broad range of career opportunities if you want to get into archaeology, and it's pretty diverse.
All right, thank you so much.
You're welcome.
All right, as for final questions, well, thank you again. If you can hang on for just a couple minutes, just in case there are some questions, or if not, we can make sure you get those through your contact information. But thank you very much. All right, so for our next presenter, it's Chris Rao. He is going to be talking about neuroscience and injury. Are you there, Chris? Hi.
Hello.
So let me go ahead and make sure you can share your screen. Sorry, one second. Here we go. All right, so without further ado, I'll let you take it away.
All right, thank you so much. Let me sort this out really quick.
Take your time.
Can you see my screen okay?
Yes, I see it.
Awesome. All right. Thank you for having me. It's so nice to meet you all. All right, so my name is Chris Rao. I'm an assistant professor, actually not in Kentucky, but over in Virginia. But I am a professor in Department of Basic Science Education in the medical school that's here at Virginia Tech. So I teach primarily the first and second year medical students about neuroscience. So we'll delve a little bit about that for today's talk. So the focus, of course, will be spinal cord injury recovery. All right, excellent. So in the next 20 minutes or so, we're going to cover a pretty broad territory. So we'll talk a little bit about the nervous system, just kind of introduce you to some of the structures that are involved here that are really important. And then we'll talk about a really cool phenomenon called neuroplasticity and how your nervous system changes throughout your life. And then we will also, of course, talk about treatments and therapies specific to one component of this nervous system being the spinal cord itself after injury. All right, so this is a image of your average neuron. There's a lot of different morphologies and structures of these neurons depending on their function as well as where they're located in your body. But you have 86 billion of these things throughout your whole body. That's a billion with a B. You can see it has a cell body there in the center and a number of different processes. And all of those little processes make interconnections with a whole bunch of other neurons. So here's just a sample of that. This is showing calcium imaging in a mouse cortex in the brain of a mouse. You can see this type of imaging allows scientists to be able to see the electrical activity or action potentials that are produced in all of these different cells. And you can see how they're communicating with one another. They're firing back and forth and talking to one another. And that's how you control a lot of different functions in your body. So those are the building blocks. You also have, in addition to neurons, you also have a whole other group of cells that we won't get into called glia that play a super important role in your nervous system's function. Now, on a systems level, we can split up the nervous system into two primary components. You have the central nervous system, which is shown here in red. That consists of the brain and the spinal cord and also the brainstem. So these are the central processes of your nervous system. So the brain, of course, it processes, interprets, stores information, and gives orders to all the various body parts that you have. the different muscles, glands, and organs of your body. The spinal cord is what connects your brain to the rest of your body. It sort of bridges that gap to what is referred to as the peripheral nervous system, which is shown here in green. So the peripheral nervous system consists of all of the other nerves that innervate a whole different types of structures of your body. So it innervates skin and muscle, viscera, tendons, bones. Just about every single part of your body has nerves that are hooked up to it. All right. Now, with the peripheral nervous system, you can also split it into a sensory component and a motor component. So you have sensory nervous system and motor nervous system. In addition to that, there's also another component called the autonomic nervous system. And this is important for regulating involuntary body processes like heart rate, and your blood pressure, respiration, digestion, a lot of the things that fortunately you don't have to think about on a consistent basis. All right, so now we'll talk about each of these little components one after the other. So we'll start out in the periphery, so the spinal cord and your peripheral nerves. So this is an image of what your spinal cord looks like, and then there's this long peripheral nerve that forms from both motor and your sensory nerves that are coming out of the spinal cord. And then at the very end of this long peripheral nerve they continuously branch out as it's going out further and further into your body, and it'll make connections with your viscera, or your muscle, your skin. And at the very tips of those individual nerve fibers, you have a whole bunch of different specialized endings, depending on the function. Some are more specialized than others. So the lower left panel here, you can see what a motor nerve ending looks like. So this is what will make that interconnection with your muscle cells to make them contract and help you move your muscles and move your body. On the sensory side, you also have a number of different sensory endings that detect all sorts of different things. And this sample here on the lower right panel is just a bare nerve ending fiber. This is the type of sensory organ that detects things like pain and temperature. You have other types of receptors that detect vibration and movement across skin, and pressure, and all sorts of other different really cool things. It detects all the stimuli and then ships it up to the spinal cord and then up to the brain. Now, this is an image then of the component of your central nervous system called the brain. So this is what your brain looks like. These are what human brains look like. And you can see there's all sorts of really cool ridges and valleys. Unfortunately, we're not going to be able to get a chance to go into too much detail on what they all do. But one of the things that scientists have done is determine all the different functional parts of the brain. So in this, we're not going to go through all of this. There's a lot of text, I know, but just so you know that some of the different functions that are involved, and it relays all sorts of things from your senses like hearing and smelling, and you have like motor functions that are involved. There's just specific regions of your brain that are devoted to specific functions. Probably the most important that makes you a little bit more advanced than Jello is this prefrontal cortex that's located in the front. And this is important for your executive functions, like concentration, planning, judgment, that sort of thing. This is the main part that develops throughout your development. And it is also super important reason why you should be wearing a helmet when you're doing things like skateboarding and rollerblading, riding your bike, going on scooters and whatnot. All right. So let's talk a little bit about the concept of neural plasticity. So what is that exactly? Plasticity in itself is a quality of being easily shaped and molded. So you can think of neural plasticity as how your nervous system can change over time, and how it can be shaped or molded due to a whole bunch of different things. So the neuroplasticity can occur in a number of different reasons, for a number of different reasons. The one that we'll be talking about today is as a response to injury, but there's a whole other bunch of things that can happen. Also during development, your nervous system is changing all the time, especially in your brain. There's a whole bunch of new connections that are made as you're learning. you're also undergoing a whole lot of plasticity. Also, just your normal interactions with your environment, talking with your friends, listening to your teacher. There's all sorts of changes that occur in your nervous system. So basically, anytime and all the time, your nervous system is actually changing, which is a really cool thing to think about. Now, the levels at which these changes take place can occur all the way between molecular levels all the way over to the behavioral component of things. So these sorts of changes can take place in a whole bunch of different levels. And depending on the type of plasticity, neuroplasticity that is occurring, these changes can happen either slowly or rapidly over time. They can be just temporary, or they can be permanent. And if anybody is interested in the concept of neuroplasticity, this is a really cool book that talks about how the brain adapts to different changes and diseases and disorders that might occur. There's going to be a siren. I work right next to a hospital, so I do apologize. It'll be a little noisy every once in a while. All right, so let's focus just on the concept of neuroplasticity after injury. So there's several processes that can occur due to injury to the nervous system, both in the peripheral nervous system as well as in the central nervous system. And these can be split into three general categories: axon regeneration, collateral sprouting, and neurogenesis. And we're going to talk about each of these components one after the other. So for axon regeneration, you can see that this is your average cell, just an average picture of a nerve cell with a cell body. This long component called the axon is what hooks up from the cell body over to some sort of peripheral target. Either this could be in the central nervous system or the peripheral nervous system. So somewhere in the periphery, this nerve ending is located. Now, because of the axon is super long, a lot of times what gets injured is the axon itself. So if you have an injury out in the periphery, what will happen is that axon will die back, and it'll recede and protect the cell body itself. So whatever that peripheral target might be, you no longer have that interconnection. Now, over time, in certain parts of the nervous system, that part will grow back. And so you'll be able to regenerate that nerve cell, which is a really cool phenomenon. It doesn't happen all the time, and it happens mostly in the peripheral nervous system because, as we'll see soon in just a few moments, it doesn't work very well in the central nervous system for a number of different reasons. So that's axonal regeneration. The other thing that can happen is collateral sprouting. So in this example, you have a bunch of cells that are, again, innervating some peripheral target. If you have an injury and it wipes out some of these clusters, the whole cells will die. And so you no longer have those cells to regenerate. You no longer have those cells to re-innervate some sort of peripheral target. So what can happen is that some of those other cells that were left over, that were still intact and not injured, what they can do is they can sometimes regrow and re-innervate some of those peripheral targets. So initially, they didn't make those connections. But after injury, that will cause those cells to start to generate their own axons out into the periphery and innervate some sort of target. So that can happen as well. The last thing that in theory can happen is neurogenesis. So this is the creation of new nerve cells. So in this left panel here, you can see in the lower left corner, there's what's referred to as a stem cell. And that stem cell is what can grow into a nerve cell. So if you have an injury that takes out a whole bunch of these nerve cells, the nerve cells that are already present there and making those connections in the periphery, what can happen is those little green stem cells can move into that area, and they can generate entirely new nerve cells that will then grow into the periphery. Now, this does not happen, especially in humans, very often. This is actually quite rare. When you're young, you're able to regenerate some nerve cells. And this is young, I'm talking about just a few years old. As you progress through your life, it becomes a lot more difficult, and it doesn't happen very frequently as far as generating new nerve cells. So when you're my age, you're not going to be generating more than just 1 or 2 per year. There's not very many that can be regenerated. So what happens when the nervous system is injured is that there's a whole bunch of different things that can take place. And these changes can occur out in the periphery, wherever that nerve might have been injured. They'll also, not just at the site of injury, but also you'll have changes that take place within the spinal cord, as well as the brain, as far as different changes in the molecular reconfiguration, as well as anatomical and different connections that can be made. Now, in the peripheral nervous system that regenerates super well, a lot of times all you need is to kind of help guide that nerve cell. So there's some things that you can do, like grafting injured nerves with donor nerves is one way, and it'll help guide those new nerves to find their way out in the periphery. There's also a whole bunch of different conduits and scaffolds that can be made thanks to bioengineering techniques. So nice little tunnels that these nerve cells can grow through, and all these supportive cells that you can add and help them grow. Of course, we also have a bunch of neuroprosthetic devices, some that we've fortunately advanced quite quickly, especially with this 3D printing. You can now have all sorts of other cool technologies. And we're starting to develop these really sophisticated neuroprosthetic devices that can even allow the patients to regain sensation, which is really neat. So that's in the peripheral nervous system, which regenerates really, really well. And in the central nervous system, though, it doesn't regenerate very well. So any sorts of injuries to the brain, in this case, we'll be talking about the spinal cord. You can see some of these spinal cord injuries where an individual broke their vertebral column, and you can see it's pinching on the spinal cord, which is there in that line in white. You can also have bulging discs that can also impinge on that spinal cord and create damage. So what happens in these cases? Now, why does the central nervous system not regenerate well? So there's some specific cells referred to as glia that are only found in the central nervous system. And these cause a lot of problems as far as being able to regenerate and regrow those nerve cells. So in this case, if you have an injury to that axon, what will happen is that you have a bunch of glia, referred to as microglia, that'll chew up all of the injured cells. And in addition to the microglia, you also have a whole bunch of other cells called astrocytes. And they're all types of glia, but they kind of bunch up together. They produce a bunch of chemicals. They degrade all the tissue. And what forms is this scar, and that scar prevents any sorts of regeneration from being able to occur. And so that is the phenomenon that a lot of scientists have spent a lot of time trying to overcome. So this just shows the pattern from hours all the way through months of all of these different components that change, the stages that occur after some sort of central nervous injury. So inflammation, you have that glial scar that forms, and then, if there's any little bit of tissue that's remaining, some cells might be able to regenerate, but a lot of times it just is impossible to regrow through that region. So there's a couple of different therapies that have been developed to try to overcome this. So one is activity-based training. So you can see in this example, and this is research that's being done at the University of Louisville. So you have an injured patient. What will happen is eventually you're going to have some of the training that occurs. So this is trying to get these kids to actively walk and move again. At least initially, it's through physical manipulation and just getting their muscles to relearn how to do that and trying to reform those connections and that communication with the brain. And as we go along here, you can see the patient is going to be able to move on his own. You just have to get him started, and you can see he's starting to pick up his legs. We'll move a little bit further here. You can see with this, and this training takes place months and months, and these kids and adults, they have to devote a lot of time to do this. But over time, you can regain some function this way. Another concept is epidural stimulation after injury. So the idea of this is that after an injury, you have that interruption of connection between your brain and a part of your peripheral nervous system. So here it'll show in just a moment. See this little spot in red here. That signal from the brain is no longer being able to move to whatever is injured. But these circuit boards that can be inserted, these epidural stimulators, can bridge that gap between what is injured and what is not injured and send that signal on down. Now, this technology has developed in the last 15, 20 years, especially in large part by efforts at the University of Louisville, which is really cool. This is a recent report that just came out, and this man who's been paralyzed for 39 years is now able to stand thanks to this technology. And it helps all sorts of other things other than being able to walk, a whole bunch of other components of quality of life that are really important. And there's other research that's being done that combines not just the epidural stimulation, but also all of the activity training that occurs. And the combination of those two seems to be helping a lot of these patients regain this quality of life that they were missing due to paralyzation. So there's a lot of really cool technology that's being done. So that was super quick, but thank you for listening. I appreciate it.
That was awesome. So I do have, there's a couple questions here. The first question I see, if someone were to cut open, I guess the brain, if someone were to cut open the brain, what would be the visible differences between nerves and veins?
So the veins they'll be a different color for one thing. If you look at the nerves, they're sort of an off-white color. Veins, if it's a living person, there'll be blood flowing through it, so they'll have like a purplish color to them. So the blood vessels are a lot, unless you get down to like the capillary level, like super thin, like the blood vessels are a lot larger than the nerve cells are.
And I know you talked specifically about like injuries, but what about, there's a question about strokes. Like what happens, when you have a stroke, is it something similar that can regrow nerve damage, or is it something completely different?
Well, so it causes damage. So a stroke, depending on what it is, you can have like a blood clot that prevents blood from flowing, or the other is a hemorrhagic stroke, in which you have a brain bleed. So strokes involve the vascular system, and that disruption, if the blood is cut off from parts of your brain, that means that your brain's no longer getting a lot of the oxygen and the nutrients that it needs to survive. So there can be some damage caused by that. That being said, the nervous system is super good about being able to reinnervate these areas. It's a lot more functional when you're younger. It regenerates a lot better, but your brain can overcome a lot of these injuries that can occur, including due to strokes.
Excellent. All right, we have another question here. Could we use the technology from epidural stimulation to heal other parts of the brain?
Yeah, similar concept. Yeah, actually, there are ways, the ways that are being developed of inserting, basically it's like microcircuitry, like little circuit boards in different parts of your brain to help you regain some function. So some of that sensory function of people who have amputations, there's been research showing that has used that as a way of regaining sensation of those lost limbs. There's also ways of little circuit boards that they're hooked up to our language centers that are able to kind of measure the brain waves. And for people who are no longer able to speak, if they had some sort of stroke or some sort of paralysis that prevents them from being able to move their jaws and speak, the circuit boards can help interpret what they're trying to say. So there's a lot of really cool technologies that are being developed. The brain is super complicated, and there is so much that we don't know. So if any of you are interested in becoming a neuroscientist or a neurologist, there is absolutely room for you, and we certainly need all the brightest brains that we can get to help us.
Excellent. So another question here. Can we use this technology to treat neurological disorders?
It depends on what the disorder is. These techniques are mostly helpful for any sorts of regeneration. There's a lot of molecular therapies that are being developed to try to help treat some of these neurological disorders. There's a lot of different therapies, things like multiple sclerosis, where you're losing some of the, it's referred to as the myelin sheath, that helps speed up these action potentials. There's different therapies and pharmaceuticals that are being developed that try to help with recovery of some of these neurological disorders and neurodegenerative disorders. There's a lot that we don't know about the nervous system, like I said, and a lot of these neurological diseases, like Parkinson's and Alzheimer's disease, that we're still trying to develop therapies for, and particularly for patients who are already developing these disorders.
So I'll open it up for some final questions here, but I do have a question for you. What got you interested in going this path studying the brain and injury?
I've had a lot of family members who have had neurological disorders like Alzheimer's disease. My father-in-law actually had multiple sclerosis. There's a lot of close connections of my family that have all these sorts of disorders. But I really was, as a college student, I was in microbiology, and then in graduate school, you have the opportunity to further explore your interests in science, and I just settled on neuroscience. So it's just a lifelong curiosity about biology in general, and science, love science, all through high school. That really got me interested, and just every step along the way, you gain new experiences in research, and just the people that you interact with, that leave a big mark on you. And that's just going through life, I settled upon neuroscience.
That's great. It is amazing to see how much the field has changed, and so I'm very excited that you were able to share that with us. So thank you very much. If you want to stay on a couple minutes, if you can, just in case there are questions that pop up. But thank you. So I'm going to be introducing our next speaker here. So Dr. Kolmann, he's going to be talking about concrete dust and how that can impact our health. Are you there?
Yes, I'm here.
Hi. So let me make sure I can give you permission to share your screen.
All right.
And then take it away whenever you are ready.
Right. Thank you so much. Let me try to share my screen. I'm going to find it somehow.
Take your time.
Okay, basic. So, all right, let me do differently. The thing is, I have maybe too many.
Oh, yes. It's a lot of moving parts putting on a program, so take your time.
Yeah, so let me see this one. Can you see anything so far?
I just see you. I do see you, but I don't see a presentation or another tab.
All right, let's see. Can you see anything so far?
No.
Nope. Okay. All right.
Oh.
Can you see anything different?
So I see the what's hiding in concrete dust. So I see the
main slide?
so it's in the PowerPoint view. It's not in the presentation view, but I do see it.
All right. That is complicated now. Still not in the presentation mode?
Not yet. All right, it's larger now, but it's still in the PowerPoint mode.
So it looks like I'm sharing the wrong screen. So that means I'm going to stop sharing and then share the right screen. Sharing, so the right screen is this one maybe. So what do you see now?
I see just like a computer background. It's okay. Are you using multiple monitors? Could that be part of
Yes,
it?
yes. I'm using multiple monitors because so I usually have 2 monitors, and then now to have the camera, I need a 3rd monitor. I mean, I need the laptop monitor now, so that's what's giving me all the trouble. So maybe I'm going to just stop all the monitors and use only one.
If you'd like, you can send it to one of us and we can share the screen if you would like to do that.
I think is, let me see. So if I use just, that is crazy.
That's always something with technology. That's always the case. So
Let's try this again.
Okay.
Let's try this.
Okay. So again, I see your computer, I see your mouse. And now I'm seeing you looking like you're opening up a document. So now it's the PowerPoint. You just need to go into presentation mode.
You see a presentation mode? Nope. Okay.
I do see the PowerPoint though. Yeah, so go ahead, go open up your document again.
That is crazy. So what do you see now?
I just see your computer like background. So I see the document, what's hiding, and I see your mouse.
Okay, so I'm sorry for this. It's taking too.
We will figure this out.
Crazy. Okay, so let's see. All right, sorry. What about if I do this? Open from here. So what is this one? This one is to identify this one. Okay, so let me try again. Here, share. I'm trying to share the screen too. So I'm trying to share this one. Share.
I see your computer.
What about now?
I still just see the computer.
There's something wrong here.
I've
tried all the possibilities I could think of. The only thing that I need to do is probably just to disconnect all the thing and stay with only one monitor. What do you see?
Now I just see you. I don't think you're sharing your screen anymore.
Not? Okay, so let's see. Now I think should be better because I have only one monitor. So this one share.
Yep,
What-
it's perfect.
All right, I'm sorry about this.
Oh,
The solution was easier than I was trying to figure out. So, yeah, so today we're going to be talking about, it's kind of the end of the conference, so let's play some hide and seek. So the question we are asking here is, what is hiding in the concrete dust? So my name is Joseph Calvin Kolmann, and I go by Calvin Kolmann. And I'm glad to be here. Thank you for inviting me to talk about this project that is still in its infancy. So be patient a little bit. So the next, okay, now, so who I am, I was born in Cameroon. So this is the world map, as you can see. I was born somewhere here, not exactly that point, but somewhere there. And I went to Germany after my, to do graduate school to have my PhD. Then I moved to Sweden for my first postdoc before actually coming to the US. And I came to Louisville, so I've been here for the whole time in 2006. So usually when somebody asks me, where are you from? I say I'm from Louisville, but they don't want to believe me. So I have to say that I'm from Cameroon. So you have my background. I'm a married person with 5 kids. And right now I'm assistant professor at the Department of Pharmacology and Toxicology at UOL. So let's go concrete. What is concrete? We are talking here about the name, and it is a very complex mixture of many things. You have some aggregates like tiny stones, you have rocks, you have fine aggregates, you also have air in there, but most importantly, you have water that help to get the mixture. But maybe one of the most important component of this concrete is cement. And that's what we're going to be talking about mostly today. In addition, you have things called admixture. That is just to change the property of the concrete. For instance, if you want it to settle fast or to settle slowly, you can add a different kind of admixture. So concrete is used in basic foundations. So every house has some concrete in it. It can also be used for wastewater treatment in wastewater treatment facilities, or as well water treatment facilities, in parking structures, floor construction, exterior surface, and so on. So none of us can say they haven't seen anything with concrete today. So we are interested in cement, as I told you before, and the most famous one is called the Portland cement. So I'm going to try to show you here how this cement is produced. Cement has really 3 main components. So you have the clay, you have limestone, and sand. So with an excavator, you're going to harvest all of these components and put in a truck that will go to the factory, and the limestone will be crushed twice. So first, just like a rough crush, then you get a fine grinding, and at the end, what you have there will be mixed with clay and sand in this equipment here. Then an important process during cement production is that you're going to heat the whole mixture. So it first goes through a preheater, then end up being heated here at very high temperature, and you're going to have some clinker that will remain, but your cement will be actually collected. So is it during this process here that actually you have most of your dangerous chromium being generated? I'm going to come back to that. So in cement, you are interested mainly in the hexavalent chromium, also called chromium-6. So chromium exists in 2 different forms that are stable. I mean, in many forms, but 2 of them are stable, namely chromium-6 or hexavalent chromium, and trivalent chromium, which is also called chromium-3. So when you have such high temperatures, like above 1300 degrees Celsius, you end up creating a good environment that will oxidize all the chromium-3 into chromium-6. So basically, whatever you had in lime or all these ingredients, you end up turning mostly into chromium-6. The problem is that this chromium-6 is not good. It is very dangerous. I'm going to explain that a little bit later. So the chromium here we originate from raw material or fuel, or also from the Manisa chrome kiln factory brick, and of course also wear metal from raw mill grinding process. In Portland cement, the chromium level range from 1 to 30 ppm parts per million, and in the US cement samples, you have about 0.225 milligram per kilogram of sample. In concrete, it's a little bit higher, so 1.27, and in Icelandic cement is even higher than in the US cement sample. So what is chromium? This is like a summary of its identity. So this is like a headshot, you could say. You have the atomic number right here, 24. The symbol is, of course, Cr, and it belongs to this group of metals that are called transition metals. As you can see here, this is chromium, and its atomic weight is about 52 grams per mole. So the density is 7.19, meaning that it's way heavier than water. So another thing I should mention here is that hexavalent chromium is the most toxic one, but also those that are particulate are even more toxic than those that are soluble in water. So what is hexavalent chromium? I'm going to talk a little bit more about that now. First of all, the name chromium is just from the Greek word for color, that means chroma. And the exposure to hexavalent chromium is really widespread. So it has been used in industry for over 2 centuries now, and is used as a pigment, as a rust inhibitor, or even antifouling agent. It can enter air, soil, and water from many routes, and it is estimated that more than 170,000 tons of chromium-containing waste are released each year. So this means this is a very important environmental problem. So where do you find chromium? I'm sure that many of us have driven today or were aboard a car, and you have seen this pavement is actually made with chromium. So this is just a nice sample of colors that you can have with chromium. And here you can see electroplating industry as well as welding. They all generate a huge amount of chromium. So these people working in such factories, they're actually exposed to enormous amounts of chromium, and it's also an occupational problem. So in summary, we can say that the hexavalent chromium is a human toxicant and carcinogen. So it actually causes cancer. We don't know exactly how, but it has been shown that the chromosome instability is part of the mechanism, as well as inflammation. The exposure, as I mentioned briefly, can be natural and also anthropogenic. So it can be human-made. And so in this case here, you also could say that occupationally, one can be exposed to chromium, but also just in the environment. And this, we are talking about millions of people exposed every year in the world. And it is known that chromium causes lung cancer and also some reproductive issues. And this is based on the epidemiological and animal and cell culture studies. And I really mentioned already, the particulate form is the most potent of this. Normally, the trivalent form, that is the other chromium that is stable in nature, is not carcinogenic. So we worry more about the hexavalent chromium. So the picture that you see here is actually taken straight from the website of Advanced Ready Mix. And of course, when you see this, you can see a lot of activity going on. You can also be confident that you can be, you can have your product and everything. You see all of this. But you can also notice that it's very difficult for you to see anything here, like in the sky. You cannot see the sky here. And just because of this massive dust that is being produced here, and during all this concrete making process. So is this a problem? I think most of us will say yes. And at least the neighbors of this company see a lot for themselves. They are first of all worried about dust settling on their stuff on the backyard, or maybe sometime get in the house and so on. But more than that, we are seeing chromium 6 in this hexavalent chromium, and which means they can also have some health issues. So this is the map of where this company is. This is where I am right now talking. And we are talking about a few blocks just from that company here that's called Advanced Ready Mix. But of course, the population that is really immediately concerned is the neighbors here. And this neighbor is called Butcher Town. So if you zoom into this, you're going to see that actually this company, they have some extension here. So they have not only that one, but also this. And even getting the matter worse, they have a tanning industry here, the Tasman Industries, that actually is dealing with leather. Usually they use chromium to also color the leather. So there's also a possibility of having to deal with chromium pollution from this company. So, as environmental scientists, we need to learn more, but the question is always, how can we do this? The first thing, of course, science, you can observe, you can see what's happening and try to make some conclusions. But in this case, it's going to be tough, just looking at that dust and then say anything really serious. So what we need to do is, first of all, collect environmental and some animal samples from there. So we could collect like air, water, and soil. And from this one, we can try to find the amount of the chromium that are in there. And since concrete dust is really the main problem there, we also want to collect some samples from that. And our biotic samples will be pillbox mainly. And not that also mosses are found in that area as well, but we are just collecting pillbox. The second thing is that we need some protocols that will actually help us to do this. So maybe not so obvious, but we need to have a protocol of sampling, because when you are going on a field trip, you really want to have everything that we need. You don't want to get there and realize that, oh, you forgot a beaker, or maybe you forgot a tube or whatever. So you want to have a clear plan, what you want to do, so that when you are on the site, you just do the work and collect the sample and leave. And of course, we need a protocol for chromium assessment. And last but not the least, we need to engage the community in Butcher Town to help, because sometimes you may want to collect samples from their backyard, or from their porches and so on. So let's talk a little bit about roly-polies. So roly-polies are some cute isopods, very small animals. And the characteristic of this is that whenever they are stressed or feel like in trouble, they're going to make themselves like a ball. So that's what in their name, poly, but actually they have a very long name that is Armadillidium vulgare. So these guys here, they are very important in trying to figure out what's happening with pollution, because they can bioaccumulate metals. So they do this. If you look at this figure here on the right, they do this using this organ that is called hepatopancreas, which is also serving as a digestive organ. And so they're going to accumulate the metals here in two different kinds of cells. So one cell here is called B cells, and the other one is called S cell. So the S is just for small, while the B is for big. And the thing here is that the S cells actually help store the metals, so they can stay for a long time, while the B cells are mainly for excretion, so they secrete the metal that they have collected, and they also renewed very frequently. So trying to play with these two kinds of cells can actually help us to determine, roughly if the pollution is something just ongoing, or if it's something that has been there for a long time. So this is what we do concretely in the field trip. This here is not really playing hide and seek. I mean, you can't see the person in here, but there's somebody here that is trying to find the roly-polies. And you can see here our friend Luz, who is also the coordinator of the Community Engagement Corps of the CIEHS, trying here to get some roly-polies from the dirt. And so when we go to the lab, we have settings like this. So we have two sets of petri dishes. So one set we have paper towel like this, and then the other set we have no paper towel. So the paper towel here is to try to purge the samples, and purging means that you're actually getting rid of the B cells because they renew very frequently. So by doing so, we're going to have the roly-polies that will also have, that will mainly have S cells, and we can see really what is stored in them, not just the total amount. So you can check the total amount with unpurged samples that we also have without paper towel, and then we can check the other one that are purged. And so this is just showing you how they look like when we collect them in the field. They are very small. So here you have a bunch of them. But what you should know is that usually per site, we want to collect about 20 isopods, if they are big ones or even more, because we are trying to have enough so that you can actually analyze the sample. So in this case here, based on the wind direction and the time of the year, we target to collect that sample north, east, and west of the ARM facility. And then the half of those animals were purged for 2 days, and the other half was kept on substrate. So what we call substrate here is whatever you collect them from. So if you collect them from here, we're just going to get some dirt out of that too. And then in the lab, we're going to keep the animal on it. So they constantly have their B cells. And so they constantly have everything. They just behave like they were still on the site. Then what we do to analyze is that we're going to digest these animals in nitric acid for about 75 minutes at 95 degrees Celsius. And then we're going to share through a needle. So you're going to try to homogenize them, pass through the needle and then filter the samples. After we filter the sample, we're going to adjust the whole nitric acid to 14%. And analysis will be performed by atomic absorption spectroscopy. So just briefly, these are the results that we have so far. We have done 2 major field trips. So the first one, we just went there just to really take the temperature of the place. So we had just taking from the concrete and then having, so this is the concrete that are on perch. So this, they have their B and SS, while this only have the SLs. And you can see that in general, you're going to have the on perch amount more than this, meaning that the animal were still getting some of the chromium from the place. Also here, we didn't show the control, but we took another sample on a tree just close to in the facility actually, just a few meters from that side, and we didn't detect that much chromium from it. And so you can, in our second trip now, this is what we had. So in the facility itself, we had very high amounts. And here, in this case here, perch and non-perch was really almost the same. Probably it was very high too. So, but when we went a little bit further from the facility, as you can see, the amount are a little bit less than what we actually have there, although you can still detect some chromium on almost all the sites. So this one is the oak tree that was in the facility. So we see that you have chromium there, but to a very lesser extent. So how do you have this? The dust can get to the tree. You can also have the bugs that maybe crawl from the concrete to the tree later on or so or whatever. But the truth is that this just tells you that there's an environmental problem, not just directly related to the to the concrete itself, because the life there is really in trouble. So this is the just very preliminary result that we had. This is our team when we went to the second field trip, and this is Johnny. He's our lead scientist on this project. This is Jamie. She is another colleague with whom we are working on this project, and this is myself. It was a little of cold, and I react to cold always differently because of my origin. But yeah, this is Luz, this is Josie, and yeah, Sarah, and I think this is Jamie too. Yeah. So this is the group, and I'm glad to take any question from you, and I would like to thank the WISE Laboratory of Environmental and Genetic Toxicology, and also the CIEHS, especially the Community Engagement Corps. So these are all the people I already mentioned, all of them, except for Dr. John Weiss, who is the head of this lab, and Denifer Toyoda and Idoya Maiza, who are graduate students in our lab. Thank you so much. I'm going to take some questions if you have time, and I apologize again for my tough beginning.
Okay. Thank you, Dr. Kolmann. We do have a few questions for you. The first one that we have listed is, are there any regulations about chromium content? Yeah, absolutely. There are regulations, and because it is a carcinogen, a lot of so when they say there are regulations, it's actually when it comes to the concrete factory like this one, because these people actually have been cited many times in the past, for trouble, but nothing is really done. I mean, they haven't changed. When you get to the site, the only remediation that you see there is that they have a sprinkler, basically just to remove dust from the cars that are passing by, and sometimes also this sprinkler causes more trouble, because if you are just in the neighborhood, you're going to get some dust on your car just because of that. Anyway, there are regulations, but the issue is, are they really apply, and especially in this case, that the answer will be not that much. No, that makes sense. There's another question here: is what made you want to investigate the chromium levels? So the chromium, we are investigating chromium level because we know that chromium is a carcinogen. We know that there are people around that place that may be having some chromium. I mean, we cannot say for now if what they have in their backyards or in their porches or whatever is actually enough to, to make them sick. But we want to know,
we want to know if they are in trouble and what is the level of trouble. If it's only the dust without that much chromium, it's a different problem. So we need to know for sure that this community is not in danger.
Excellent. I do have another question. You say you talk to the community. How's the community's response been to all of your research you're trying to do? Have they been responsive, or what's that process like?
Right. So in our plans, we are going to talk to the community. We haven't done it yet, but it's something about the community. It's not. I mean, for us, it's about the research and curiosity of researchers, but for them, it's about their health. So we want to engage them, and we want to try to do our best to actually help them to tell us about their feelings and everything. But you should also know that they have that neighborhood, the Butchertown neighborhood, has actually had a whole discussion, complaining about this. So it's not like something that they are happy with, So we believe that they're going to end up helping us, and to help them basically.
Excellent. All right. This is a good question, especially for anyone who's been very fascinated by your presentation. How can a person enter this field of environmental health science?
Well, the short answer is contact me. But the other answer is that actually in U of L, we have a lot of teams that work in environmental science, health science. So not only you have this CIEHS that is really expanding. I mean, this like the second or third year or so. And this is a massive undertaking. So if you contact anybody at U of L, or we can help you how to start. But if you are a high school student or undergraduate student, and you want to, join a lab, you always have opportunity during summer to engage you. So you can come and see what we are doing, and if you like it, you can stay, or you can change your mind, whatever. But yeah, there are some possibilities for that.
Excellent. Now we have kind of another follow-up question to the regulations. So if regulations aren't effective, what can we do to protect our neighborhoods from chromium levels?
That's a tough question. What can we do? I think the first thing for us is for these people to be aware that the dust is not just not just dirt. It's not just something that is bothering them because it doesn't look good. The first thing for us as scientists is to raise the awareness that in this dust you might have something that can harm you. And then if they already have that, they can try to avoid it more, because I forgot to mention that environmentally, what you get chromium is that you inhale. So actually it's by inhalation that you get problem with chromium, and then you end up having lung cancer. So if, for instance, people could maybe wear masks, I don't know. I mean, we are in COVID area, so it's not a news, it's not a new thing anymore. But there are ways that people can try to mitigate if they actually know that there's a problem. And our role as scientists is really just to raise that awareness, basically.
Excellent. All right. So we do have another question that just popped up. Do chromium levels affect certain communities more than others? Example, lower income communities or marginalized communities?
I won't say that, because it depends on what is there. So basically, if you have, let's say you have a company that is dealing with chromium in your environment, in your community, then you are exposed to that, in your neighborhood. Now, if you are talking about people working in different factories, it's tough for me to determine if what kind of people are working in which kind of company, because it depends, really what they are paying. So I think that is a tough question, but it really has too many variable inside that I cannot, fully address. What I just can say is people working in companies like electroplating, like tanning industries and so on, that deal with chromium every day, they are more exposed, and they should, be more careful. So that's all I can say.
Okay. Well, thank you so much. I'll leave it open for any other final questions. I guess the last question I have is what got you really excited about this field?
Well, so first of all, when I grew up, I used to feel they say, what do you want to be in the future? I say, I want to be a teacher or a doctor. So basically, you can see today that all of them failed because I'm no teacher, I'm no doctor, according to my scale at that time. But when I realized in undergraduate that I couldn't go to medical school because I was in Cameroon, I was trying to get into school, it was not easy. I decided that I'm going to do something for health. So even if I'm not, directly responsible for people's health day to day, but at least I can do something either to help them to prevent disease or to cure disease. So my research life has actually spanned a lot of things, and the common theme is just helping to improve health. Actually, I'm new in the field. I started this only about 1.5 years ago. And the thing is that I was just, charmed by the massive involvement of U of L in this environmental science. But I've done a lot of things. So right now, what I'm doing is not just trying to see what chromium is doing to people. I'm also trying to see what I can do to prevent chromium issues. For instance, right now I'm studying beet. I don't know, beetroot, right? So the red beetroot. I'm studying that to see if we can somehow alleviate chromium toxicity in cells and animals. So, those are my plans, at least.
That's awesome. Thank you so much, Dr. Kolmann. Thank you to all of our panelists and our keynotes. Again, if you missed any of this, we have this on our YouTube page. We will also share out contact information from all of our presenters. And if you have any questions, you can either relay them to the Science Center or directly to them. Again, thank you so much. It is a little after 11. So thank you for your day. Have a great rest of your day.
Thank you. Thank you so much. Thank you.
Thank you.
All right.